We're going to have 4 speakers today, so we'll have 4 short talks, and then we will have a longer period for everyone to ask questions at the end. If you are asking questions, we ask you to put them in the Q&A as opposed to in the chat. And there's also a chat open for those of you who might like to share resources or other things that are not questions during the course of the webinar. Okay, it looks like our number of participants is stable, so I will get started, and if people are still joining late, that's fine with us as well. So in today's webinar hosted by FENS, we are going to be talking about visualization, and specifically we're going to be focused on creating clear and informative images for scientific publications. And we have 4 different speakers, each of whom will look at the question of visualization from a slightly different angle. It is a short webinar today, so we won't be able to cover everything that we normally would in detail in our individual talks. But we know that there is an extensive resource list that we provided online that will allow you to get more information on some of the topics that we're addressing and cover some additional content that we wouldn't be able to get to today. So my name is Tracey Weissgerber, and I am a meta-researcher. I study the science of science at the Quest Center for Transforming Biomedical Research at the CHET in Berlin. Okay, so I think before we start off any webinar on visualization, it's very important to talk about why we're here. So there are so many things that go into the science that we do in making it rigorous and robust and transparent. So why should data visualization or data presentation be on the list of things that we think about? And I would argue that data presentation is especially important because it's the foundation of our collective scientific knowledge. So when we think about how we structure a paper, very often the figures that we present are the place where we put the most important and the most interesting findings in our paper that we really want the audience to pay attention to. And unfortunately, the raw data underlying those findings aren't usually available. And that means that the figures are really all that we know and all that we will ever know about that data that was collected. And so if we are systematically making poor choices about the types of figures that we create or the way that we share our data or how transparent our figures are, then we impair our readers' ability to understand this data set, not only at the time we publish it, but also for all of the future. And no one wants to be responsible for ruining the future. So it's really important that we think carefully about how to do good data visualization. Okay, when people are just starting to make their graphics, there are 2 common but incorrect assumptions that they make. So I want to take a minute just to highlight these 2 incorrect assumptions. And the first is that readers often assume authors often assume that readers are reading the paper in the order in which it was meant to be read. So they start with the title and abstract, and then they read the introduction, and they carefully review the methods, and they've done all that before they get to the figures. And unfortunately, we know that reality is very different. There are many editors and reviewers and scientists, and especially senior scientists, who say they go to the figures very quickly. Sometimes after only reading the title and abstract, or maybe the title and abstract in the end of the introduction, and to figure out what the study is about, and then they go straight to the figures. And there are a lot of journal websites and search engines that cater to this. So for example, journal websites or search engines like PubMed will show you the title, the abstract, and then all of the figures in the paper right below it. And so it's really important that your figures can stand on their own and that they convey a really clear message about the methods of your paper, because your figures are part of how your readers are deciding what information they're going to look at and whether they're going to take their time to read the rest. The second thing that people assume is that if they can interpret the figure, then their readers can also interpret the figure. And this is also a really terrible assumption. When we're designing figures, it's really important that we design them for our audience and not for ourselves, because our audience is often very broad. So it might include scientists in our own field who have a different area of specialty or expertise, or scientists in related fields, scientists that use similar methods but study different conditions. We know it includes reviewers and editors. It might also include grants officers, educators, and patients. And all of these people have a different scientific training, different vocabulary, different background. So they may read your figures quite differently from the way that you read them. And things that are clear to you can be very confusing to someone who has a different expertise, which many of your readers certainly will. And so designing your figures with your audiences in mind is really important to making sure that your paper is clear and accessible to as many people as possible. And here's an example of this. So I want you to think about how much information you would need to interpret this image. And so if you happen to be a researcher who works with mouse pancreatic beta islet cells, you might know that that's what this is. And you might know what the dark and the light represent and what all these little structures are. But most of us would probably say, I have no idea what this is. So that unannotated version really doesn't tell me anything. The next version has some annotation, so it tells me where the nucleus is, where the peripheral insulin secretory vesicles are. I can see that these dark structures here are mitochondrium, and that this is here is cytoplasm. And that gives me some orientation to what parts of the cell I'm looking at and what features might be important. And then I have another image here that has all kinds of annotation and all sorts of features noted, and so many features that it's a little bit difficult to tell what's going on here. And so this is maybe too much information and more than I would want to really understand what's going on, but it depends on the research question that's being asked. So as you're designing your figures, you always want to think about, what does my reader need to understand my research question? And remember that your reader is not you. Okay. Now that we've talked a little bit about the general theory underlying visualization, I'm going to move on to some very common visualization problems, and we'll discuss how to identify and fix those things. So the first of these problems is that we want to look at papers that are using bar graphs of continuous data, and it's very important to replace those graphs with more informative graphs, like dot plots, box plots, or violin plots, that show us the sample size in the case of dot plots and the data distribution in the case of all of these graphs. So why is this important? Many of you who work in science will know that bar graphs are a very common way of presenting continuous data. And we've done studies showing that approximately half of papers that have a data figure have a bar graph of continuous data, which makes this the most common data visualization problem. So why is this a problem? If everyone's doing it, then why shouldn't we use bar graphs to present continuous data? One of the reasons for this is that many different data distributions can lead to the same bar graphs, and the actual data may suggest different conclusions from the summary statistics alone. So here I have a bar graph on the left, and then I have 4 different dot plots of different data sets that would give me this same bar graph on the right. And the first thing I can see here is that the sample sizes are very, very small. There's a lot of uncertainty in this data set, and I shouldn't be too confident in the conclusions. But if I look at the first figure, I see that group 2 has a bit higher values than group 1, and this might be something that I'm interested in. In the second data set, C, the higher values in the second group appear to be driven by an outlier, maybe less exciting to pursue. In the third graph, I see a possible bimodal distribution. My sample size is too small to tell, and I would need more observations to know whether this is in fact bimodal. But if it is, I would want to know whether there's some other variable that I need to capture in my data set that explains this difference. So are men consistently having lower values than women? And do I need to account for sex in my analysis? And in the last case, I have unequal n. So the high values in the second group could simply be that I've underestimated the range of values because the sample is so small. And finally, it's important to note that we can't distinguish between these scenarios based on p-values alone. There really is no substitute for seeing the actual data. The next question that I always get is, can I still use a bar graph if I know that my data are normally distributed? And thank you for asking, but the answer to this is unfortunately no. And there are two reasons for this. The first one is that bar graphs don't allow you to critically evaluate the data. So I can't see the distribution. I can't see how many data points there are. There's really no information here. It's just a visual summary of the summary statistics for the sample. And the second thing is that the bar graphs focus our attention on the height of the bar instead of what's really important, which is how the difference in mean compares to the variability in the data, or how much overlap there is between my groups. And the bar graphs are also going to distort our perception of the range of values. So when I make a bar graph, I very often cut off the y-axis a little bit above the highest error bar. And when I do that, I may exclude values from my axis that are actually present in my data set, and we can call that the zone of invisibility. In some cases, 0 or the values around 0 may not be biologically or physiologically meaningful. And so if that's the case, I can also end up dedicating a large portion of my graph to values that never actually occur in my sample and just aren't possible. And if this is happening, it's called the zone of irrelevance. And so we have this distortion of what the range of observed values actually is with a bar graph as well. So what should you use instead? Really important question and topic, and unfortunately, I don't have time to talk about it today. However, I do have a Twitter thread which tells you everything you need to know, as well as a couple of papers. So if you look at the list of webinar resources, then you'll find a link to a free webinar that provides much more detailed information and goes through the Q&A in words. And you'll also find links to the 2015 paper that we published, as well as the updated 2019 paper that goes into more detail on solutions. The next thing we want to look out for is CHET or rainbow color maps. So if you see these, it's important to replace them with colorblind safe alternatives. Why does this matter? Well, the reason is that rainbow color maps create contrast where none exists and visual distortion. So here I have 3 different pictures of the Mona Lisa. One is the normal version, the second one is in a rainbow color map, and the third is in a colorblind safe and perceptually uniform Viridis color map. So what do I need to know here? Well, if I were looking at the rainbow color map version, I would conclude that there's something crazy going on with her chest and her face. And the reason I would conclude that is because my brain reacts much, much more strongly to the reds and oranges than it does to the blues and the yellows and the green. And that lack of perceptual uniformity is what gives me the impression that there's a strong effect when there really isn't one. And then the additional problem is that these color maps aren't colorblind safe. So we need to get rid of the rainbows and replace them with Viridis or Cividis or better alternatives. The next thing that's important is to make your figures accessible to people who are colorblind. So up to 8% of men and 0.5% of women are actually colorblind. And that means that depending on how your colors are set up, they may not be able to get any information out of the graph that you have. So here's an example of a lasagna plot in red and green, and you can see that if I had the most common form of color blindness, this bright box shows what I would see, and I would see no difference between the two. So the meaning of the graph is completely lost for me. So it's important to choose colorblind accessible colors and figures, as well as annotations. And if you're not colorblind, you can use free tools to simulate what a colorblind person would see. And Color Oracle is one example. It's a tool that you can download in about 30 seconds, and then you click a button on your mouse and it will change your screen colors to simulate what a colorblind person would see. The next important point is to include a flowchart in your paper. Flowcharts are really important to help readers understand both your study design as well as assessing the risk of bias. So what do I mean by that? Well, this is an example of what a flowchart might look like for an animal study. And the really important thing with flowcharts is that they tell readers exactly when you excluded observations from your study and why. And that why information is really important to understanding whether there's potential bias in your study. So if you're studying an animal model versus a control condition, and a lot of your animals were excluded because the model was too harsh and they ended up dying before you could measure your outcome, then maybe you have some survival bias where you're only looking at the healthiest animals who were able to survive the model or the intervention. And so these reasons for exclusions are really important. One of the best ways to make a reporting or a flow chart, if you've never done this before, is to look at a reporting guideline for your type of study in order to find templates. And so you might look at the ARRIVE guidelines for animal studies, the STROVE guidelines for observational studies, the CONSORT guidelines for randomized controlled trials, and the Equator Network website lists many other guidelines for other different types of study designs that may include flowcharts. It's important to note that most papers don't include flowcharts, so only about 20% of articles that were published in top peripheral vascular disease journals had flowcharts. And again, one of the reasons that this is so important is that we know, especially in small sample size studies, which are very common, that the biased exclusion of observations greatly increases the likelihood of false positive findings in small studies. And so we want to know, if our study is at risk for bias and for false positive findings. That's really important information for readers to have. And a flowchart is one of the ways that we get that information, or that we get reassurance that there aren't any biases here and the study in fact looks good. Okay, so that is the end of my lightning fast talk on some quick visualization errors that you can identify and fix. So our next speaker will be Guillaume Rousselet, who is from the University of Glasgow, and he would like you to note that he has this lovely shirt and also supports getting rid of bar graphs. I think you're muted.
All right, here we go. Thank you, Tracey. And I'll switch to my screen. Sorry. There we go. All right, folks. So I have the T-shirt somewhere. I'm not wearing it at the moment. So I'm a section editor at the European Journal of Neuroscience, and today, what I want to do is give you some indication of the type of common issues I see in submissions related to graphical representations and how they link to statistical inferences. So when I get a submission to the journal, the first thing I do actually is, well, first thing is I read the title and then I go straight to the figures, because it really tells a lot about the quality of a paper. And I check two main things: accessibility, whether the figures are high contrast, high resolution, and use colorblind friendly palettes. And you have some example there, but Tracey went over that. And the second thing is whether there's enough information in the graphical representation for reviewers and eventually readers to make up their own mind about the data. So if I get a paper that's full of bar graph, I typically send it back immediately to the authors to be replaced with more informative formats. So, and that's the policy actually of the journal as set out in this editorial in 2016, where we strongly encourage authors to use other types of graphical representation. Obviously, the question is, what should you use? So here you have the same data represented in many different ways. There's plenty more options, and that's stuff done in ggplot2 in R. So obviously, we don't want people to use bar graph because they hide so much information. And there's always the question of what should be the alternative. I would argue that in neuroscience and psychology, where sample sizes tend to be, in most cases, be quite small, by default, really the scatter plot should be used. But it doesn't stop you from combining scatter plots with other types of graphical representations. So for instance, here is an example, a figure from a recent paper in the European Journal of Neuroscience, where you have a scatter plot superimposed on top of a box plot. So it helps give a lot of information, but you have two issues with this figure here. One is that the formatting is quite inconsistent because we have similar types of data, but yet in that figure, authors assume that their readers will understand three different types of graphical representation. You've got bar graph, line graph, and box plots here. So that's asking a lot of your reader to switch across panels. The other problem here is that there is a mismatch between the graphical representation and the analysis that were carried out, because, well, like most researchers, these authors made inferences about the population means, but those box plots don't show the mean. They show the quartiles of the distribution, which is very useful, but here on top, they should really superimpose the mean, which is what their inferences were on. And that will let you assess things like the skewness of the distribution and so on. So it's really important to try to match the figures to the analysis. But it doesn't mean that one figure is going to be enough to make sense of your data, even if it's a very small, simple design. So let me give you an example from the 2016 editorial, where we present here this 2x2 design. So we have two independent groups, and each group was tested in two conditions. So we have a classic representation of a bar graph suggesting some form of interaction. Now, it's a bit more complicated than that. So if we open the black box and reveal the underlying distribution, we could show something like the distribution of differences, condition 2 minus condition 1 for each of the group. And it gives dramatically more information than the bar graph. There's a lot going on here. But yet that's not sufficient to make full sense of the data, because we don't know the relationship between the size of the differences and the original scores. So you can do that in that case where you link the representation across conditions. And it's very important to assess things like floor and setting effects, for instance. But yet another type of representation would show condition 2 plot as a function of condition 1. And when you do that, it reveals even more clearly a pattern you can see here, in that the difference between the conditions, as shown by a departure from this reference line, tends to increase with the size of the scores in condition 1. So it suggests that there's something going on here that wouldn't be picked up by a classic ANOVA analysis. So that might suggest a switch to different type of analysis. And that's quite a simple design. And I have, if you're interested in that, there's even more detailed graphical representations you could use for even simple data sets like that. That's explained in this 2017 paper. Now, when it comes to interaction, things get even more complicated. And I would argue you need even more graphical representation. The problem is it's been clearly explained in this 1978 paper in the context of the psychology literature, but it applies to neuroscience as well. It's a very important problem that's typically ignored, in that quite often we measure one thing and we make inferences about another. So for instance, we might measure probability of recall, but we want to make inferences about information in memory. So let's say we have these results here with a 2x2 design with parallel line, which suggests that there's no interaction. The problem is that the probability of recall might relate non-linearly to information in memory. And here in that case, for that particular pattern, actually, once you express the data after that nonlinear transformation, an interaction appears. And you can have easily the opposite pattern, where you have an interaction in the original units in which you measured whatever you were assessing, and then after transformation, the interaction goes away. In neuroscience, a clear example of that would be measuring the fMRI ball signal and then making inferences about neuronal activity. We know there's a strong nonlinear relationship between the two. So maybe you make a graph showing the ball signal, and it looks like there is an interaction or not. But after nonlinear transformation, those might go away. So I strongly encourage you to read those references, because those problems are really essential. And one way to address them is to make multiple graphical representation, trying different types of nonlinear transformation to see if the interactions might appear or disappear. And that would be more convincing evidence for the presence or absence of an interaction. Other problem with interactions and related to graphical representations is a classic interaction fallacy. So I see that very often where you have two regressions or correlation scatter plot presented, one being significant, the other not, or both being significant but with different signs. And at least implicitly, the assumption is that the two differ, but there's no test of that. So here we have a mismatch between what is being presented and the inferential statistics. And that's well documented. This paper was 10 years old. I don't think things have changed that much. The other issue is the lack of graphical representations for all the results, irrespective of the p-values. So typically, people would do a whole bunch of correlation analysis or regression analysis and show only a few, typically the ones that are significant, and remove the rest. Really, for readers and reviewers to make their own mind about the data, you should show all the results, no matter what the p-values are. So for instance, in this article, the text reports 9 correlations, but only 2 were illustrated. You should really have a panel showing all the results. Now, another case where more graphical representations are needed is the famous table 1. You quite often see that in clinical studies and other types of studies where you have a lot of demographics summarized in a table like that, but it's so much data compression that it's really impossible to understand the distribution of the data and other considerations. So the alternative to the table 1 is to make a lot of graphs, lots of scatter plots showing the demographics for all the participants, or split by groups if you have multiple groups, and that provides so much more information. It's a really striking example here where you have scatter plots with box plots superimposed. And what they did here too is that they looked at their main measurements in their studies, and they expressed that as a function of other key measurements like accuracy, reaction times, or demographics. And that's really useful to have informed decisions about covariates that could be used in the analysis and so on. And finally, another place where two other places where you would need extra graphical representations are the introduction and discussion, because typically that's where people would compare studies, compare their own results to other studies. And typically this is done just based on p-values. So you can be in a very silly situation where you have 2 studies with the same estimation, but one is quite noisy, so it's not statistically significant, and the other is statistically significant, but the values are the same. So that's a case where it'd be really useful to have graphical summaries with some measures of uncertainty of the studies, or you could superimpose results from previous studies onto the graph of your own results, for instance. So to summarize, my take-home message is you need to make a lot of figures, and you probably need to make many more than you're currently doing to really make sense of the data. We just strongly urge you to share your code and your data. That will lead to a much more informed reviewing process, much more productive one, and that will in the long run increase the impact and trustworthiness of your research. And if you submit your paper to the European Journal of Neuroscience, you can share everything for free in Figshare during the submission process. And that's it for me. Our next speaker is Alberto Antonietti. He's going to tell you about bioimages, common problems, and pitfalls in publications. Thank you.
Thank you, Guillaume. Okay, so hello, everybody. In this second part of the talk, we are going to focus on bioimages, and we will see which are the common problems that we have discovered while reviewing the recent literature. But first, let me thank the FENS committees that supported the organization of this webinar, my colleagues that accepted to speak, and the whole team of eLife ambassadors that worked for 2 years on the meta research that I'm going to show. So let's clarify what we mean by bioimages with some examples that should be familiar to neuroscientists. So we're going to consider photos, and microphotos, then photos taken by means of different microscopy techniques, light microscopy, confocal, immunofluorescence, etc., electron microscopy, and medical images, so MRI, CT, PET, echography, etc. We are not talking of computer-generated images or graphs and plots. Bioimages have always had a very important role in neuroscience. In this slide, we can see the evolution of medical imaging, for instance. The first issue of the European Journal of Neuroscience in 1989 with a PET study with, let's say, quite low resolution, an fMRI study from the 2000, the images, the real scale. So I downloaded it with the highest available resolution. And today we have spectacular resources. So we are passing from images to videos. It's a 3D amplified or 4D MRI. So as neuroscientists, we really make a large use of bioimages. And regardless of the image types and how fancy was the technique to acquire it, bioimages can suffer from different problems when they are finally put into a paper and hopefully published. Image manipulation, fraudulent or accidental, is a story on its own. Today we will focus on 3 aspects that we can all try to improve in our papers and presentations, which are legibility, image quality, and accessibility. But are we sure that we have a problem about these aspects? In order to gauge the quality of bioimages in recent literature, a team of researchers from all over the world started a meta-research during the eLife Ambassadors programs of 2019 and 2019. And we consider the, let's say, top 15 journals in physiology and cell biology. A third field was plant science, but it's of less interest for neuroscientists. And we screen hundreds of papers in order to evaluate how figures with bioimages were reported. You can find the full protocol, data, and codes on OSF. Today, I will just touch some points. If you are interested, you can find the whole work on PLOS Biology. So we first looked at the types of images that were present in the papers, excluding the papers that did not contain any bioimage. And the most common type of bioimage were microphotos in 90% of the papers. Then photographs, especially in cell biology, some EM, and a few clinical and other bioimages. So considering those types of bioimages, in order to have an immediate understanding of the dimension that are depicted in the image, we need scale information. So we looked at how many papers had complete scale information reported using scale bars, with dimension written directly above or under the scale bar itself or reported in the legend. And we found several cases where the scale bar was absent at all, like here in this example, or it was impossible or very, very difficult to read due to different reasons. Low resolution, like in this second example, or where the color of the image make it blend into the background. So it was really difficult to see it in these 3 cases, or where there were too small scale bars. So if you look very carefully here, there is a scale bar. So what we discovered is that 24 and 10% on papers had bioimages without the scale information in any image. And neither the magnification reported in the legend, which is, however, an inadequate way to report the dimension of the image. And if we sum those percentage with the number of papers that just reported the magnification or missed the information for 1 or more images, then we see that almost 50% of papers resulted to have an incomplete scale information. And for the other half, with the bioimages with a scale bar, in most of the cases, the dimensions were just reported in the legend. Then insets are a very common way to show the same image at different multiple levels of detail, and you can show a magnified part of the image. And we checked if insets were accurately marked in the image and if they were described in the figure legend. And possible problems are a wrong marking, so the magnified region is not the one marked in the original image, or in many cases the inset, you don't know where the inset come from, and in some cases the inset itself cover the original image. So the insets were present in a subset of papers, but in 30–40% of the subset, they were not properly marked, at least in some images. And in 60% of the papers, they were not mentioned or described in the legend. It was in fact hard in some cases to identify insets because of the missing description in the legend. We then checked how complete were the figure legends, looking if the species and tissue object in each figure was indicated, and if the labels and annotation used were also described. And many papers have a partial description. A classic example is that the tissue or species is described in figure 1, but then it's not repeated in the following figures. And problems can arise when there are more than one object or tissue or multiple experiments. In general, labels and annotation were well described, but still in 30% of the papers, they are partially or not described at all. We have already seen that figures with jet maps or some color palettes can be inaccessible to colorblind readers, and bioimages suffer from the same problem. While there are cases where the colors cannot be changed, for instance, in a photograph with natural colors, in many other cases, the researcher can choose the colors to be used for labeling. Red and green is a very common combination. And while it's perfectly fine for a reader with normal vision, the two colors are not distinguishable if a reader suffers from deuteranopia, which is the most common form of color blindness. And it's important that both images and annotation and labels are colorblind safe. So here we can see some examples that are not accessible, where multiple colors, 2 or 3 colors, appears the same in the colorblind simulation, or where a colorblind reader cannot tell the difference between the green and the red labels. So among the eligible bioimages, there were many papers where at least some images were not colorblind safe, especially in cell biology, where microscope image with 2 or more markers are very common, in neuroscience too. And labels in general were accessible, but still around 20% of papers had problems with them. So in summary, what we discovered analyzing these hundreds of papers from the top quality journal in the recent literature is that for 50% of papers, there is a missing or incomplete scale information. That in 40% of the paper, the insets are not properly marked or not described in the legend. And half of the paper with legends not reporting the object, and the 30% of the papers not explaining the labels and the annotation used. Then 35% of papers contain figures, images, or labels which were not accessible to colorblind readers. So if we look at the papers which met all the criteria, only 10% of the papers we have analyzed met all these good practice criteria. So we can sure improve the readability and accessibility of our papers with bioimages, but how to do so? So this will be unveiled by our next speaker, Dr. Helena Jambor. She's a data visualization scientist at the University Hospital Dresden and lecturer in Berlin. And she will explain some solutions to the issues identified.
So my slides should be visible. but not in presentation mode. Is that correct? Can someone? All right. Thank you. I'm gonna be giving you 10 tips for presenting images and figures, because obviously we don't want to leave you hanging here after pointing out all the errors you could possibly make. So 10 tips for you to count on your 10 fingers, and they're also downloadable in the slides, so no worries if you miss one. The first one is, of course, as always, make an image copy of the microscopy image data file that you receive. So do never ever attempt to work on the original. Always do any kind of adjustments that you're doing on the copy only. And make sure that you save the original microscopic image file somewhere for you to go back to it later in case needed. The second most important tip is that before attempting to make an image figure, you should think about the magnification of the image and in which it will appear on the final figure. So will it be something small? Will it be something large? Will it be shown alongside many other images? So what is the size of on the final page? And in case you want to show some details that are not visible in the small image, you can think about including some kind of crop or zoom or inset. We just learned what is an inset from Albo, and please never forget again to indicate the position of the inset in the original image. The tip number 3 concerns colors, and we speak first about one color images, because not always you actually have 2 channels in your light microscopy image, for example, or you just have one color image from a normal microscopy. So what you should consider is that different color combinations have different kind of visibility. So depending on the color you choose for the channel that you're imaging, the contrast may vary of the signal on a different kind of background color. So for instance, what we see at the bottom is a blue signal. For example, DAPI, DNA stain is often shown in blue on a black background as it comes out of the microscope directly. That has a very, very poor contrast, and most people will not be able to see details, for instance, of your cellular phenotype or your intricate neurons and their boutons. So think about options that you have to show the signal with a better contrast, and the first row gives you some pointers. So grayscale is always better, and also think about the possibility of inverting the signal in the background, because dark details on a white background have the best visibility. If you do inversions, make sure you're familiar with the conversion that your software is using, because there might be some pitfalls. And how can you test whether a color is suitable and has a high contrast? That's very easy, because you can just check what it looks like in grayscale, and that tells you immediately whether details are visible or not. Obviously, sometimes we also have 2 colors in our images, and obviously you need to then still think about the background color, but one of the most important things if you have 2 or more colors is that you should be colorblind safe. And you heard it now 3 times, I think, from all of the speakers. So it is important to be colorblind safe. So make sure also that your microscopy images are visible to people with color blindness. So the above one is not accessible to every person. The below one is better. And Tracey, in Tracey's slides, for those who come later, there were some pointers on which tools you can use Color Oracle to test that. If you have microscopy images with 3 colors or even more, still try to be colorblind safe, but I will immediately point out to you that this will be a struggle and it will be essentially impossible. So you will need to come up with different kind of strategies of showing a 3-color image in a colorblind safe manner. And the best possibility you have here, if you have the space, is to split the channels by the different colors. So you have show the red, the green, and the blue channel and adjacent to each other. Or if you remember that this is not the best contrast, you actually show each channel in black and white and just indicate, for example, which channel it was in the original file. indication brings me to the step number six is that you should annotate colors. So if you made a choice about the colors that you want to be showing in your figures, absolutely be sure that you explain all of the colors that are used in your images. For example, you can show codes show the two channels inside the image directly on top of the data, or you can show it below. So this will be dependent again on the journal. For example, some do allow to show text on top of images and some do not. So make sure that you know where you will submit and what the restrictions are there. And obviously, as Alberto pointed out, this also may be a problem that the annotations are not colorblind safe. So please stick to that and also show them in colorblind safe mode. After you annotated all of the colors, and you're really good here that you have accessibility to everyone, also be sure that you annotate the scale of the image shown. So this might be provocative, I hope it's not, but we can discuss it at the end. I would say that every single scientific image needs a scale information, no exception. And just to give you a couple of indications how that may look like, you could have a white bar chart which has a fantastically strong contrast with the scale directly on top of the image. If the visibility of such a scale or the text is not great, you can always play around with, for example, having a box in the background to increase the visibility of your scale bars. And of course, if you have larger samples, for example, a whole brain or something like that, you can also include the scale directly in the image. So all of these are possible. What is frowned upon typically is magnification statements. And if you've tried that before, you probably most of the time will fail with recalculating exactly what the size of the images would have been in the original file. So let's just agree that we don't do this anymore. This is something maybe from the 50s. So now we are at step number 8, which is that after you indicated the colors and the scale used, you may also want to annotate some of the features that are visible. This is very popular, especially in dense images where intricate structures may be needed to be pointed out. And you have a couple of different options that are available to you. One option is obviously lines. They are very, very common. They've been used for thousands of years, actually, in scientific images. You also have arrows. If you are choosing to do an arrow, make sure that you use the arrows in a consistent meaning across the entire image panel. For example, some arrows could be pointing to something, as it's the case here, but arrows also are used to indicate movement or to make a call out, etc. So sometimes arrows have different meanings, and if you mix the meanings within one image panel, readers may be confused. We also have regions of interest. These are these dashed lines, for example, that can show an entire region, for instance, in a microscope image. We have symbols such as a star, etc. And we have a letter code. The letter code is really fantastic because it can, in our alphabet, label up to 26 structures, which with a very clear meaning, so we can discern them. Obviously, you have to make sure that they are explained somewhere. Ideally, all of these symbols and arrows should be explained as close to the image as possible. In worst case, in the figure legend. Tip number 9 is that you should really think about the legend. Do not just dump images onto a page and call that a figure, but think about where things are placed. So the aim for layout is always that your audience really understands the reading direction of the image, so that, like in a text, there is a flow in which the reader can go through an image panel. And 2 fantastic layouts that are nice and easy to read is that either images or images mixed together with charts, whichever is your final figure, that image are presented in either rows or in columns, and that you then go through either or. and try to avoid mixing a row and column layout. They are fantastically for confusing your audiences, and in addition, sometimes you may get it wrong when labeling stuff in your figure panels. Okay, so that leads me to number 10, a figure legend. So no image ever is self-explanatory. So we always need to add some text as close ideally as possible to the image, and if not in the image legend. And just last week, there was a nice blog from Brooke at the blog Total Internal Reflection with a lot of details about figure legend, which is why I will not comment on this further here. So if you want to go through these tips, you can go back to my slides, but also help is near to do it better. So for example, Christopher and I published last year the cheat sheets for novice users of image sync software and creating image files. So these are A4 pages that you can just print out and have next to you while you're preparing your first image figures, for instance, and one slide shows you how a couple of guides, the way that I just explained to you, and the other cheat sheet explains you how to do it with Fiji or ImageJ, so how to do it professionally. More help is near. So we have a lot of good and bad examples of what to do and what not to do in the PLOS Bio paper. So you can just download all these figures, and you will have tons and tons of examples of what to do and what to avoid. And last note, more tips are coming your way. I'm also leading a group at the Quare Blimi initiative. So this is an initiative for quality in light microscopy, and we will shower you soon with a lot more advice concerning image publications. And that is the end of my section, and now it's time for questions. And we wanted to immediately point out that we will stay until questions are answered, but obviously you can also get in touch with us later. Thank you very much.
Okay, so we have a few questions in the Q&A, and I believe there is also the option to raise your hand for those of you who would like to ask questions, not by typing, and actually say something. So if there's anyone who would like to ask a question live, please let us know. Okay, so we will start with, I will ask this question to Guillaume. Isn't the middle line in the box plots representing the mean of the sample?
By default, no. I mean, unless I've seen a few cases where people hack their code in R, for instance, to report the mean, but by default in any package, the box plot shows the 3/4, so the line in the middle is the median, not the mean.
Okay. All right. So the next question we have is from Ksenia. So the thing that often confuses me, I don't understand why researchers who applied non-parametrics to assess their data often use mean and standard error to present results in graphs. This is common. So in our 2015 study, we found that about half of authors who were using non-parametric stats presented data that they had analyzed non-parametrically as mean and standard error or mean and standard deviation. And I think this just illustrates the extent to which people are doing their visualizations on default and without training, and perhaps without also a statistical understanding. And so there's, I think in a lot of labs, it's you get your data, you make a bar graph, and it's always a mean and standard error bar graph. And then, you know, that is the way data are presented. So I share your confusion, but that's how it is. And I would appreciate your help in changing it, all of you who have attended this webinar. Okay, Helena and Guillaume?
Yeah, Helena, go ahead.
I wanted to say something to Susan Chalmers. So she's asking if the webinar is a great teaching resource and she can use it. So all of the slides from me and Antonio for sure, and also Tracey, they're available in the download material. And I can speak for myself, please do reuse them as much as you want and as widely as possible. They are CC0, so I share copyright with anyone interested.
Yeah, my slides are available too. Yeah, I wanted to add about the point of the mismatch between the non-parametric and the graph showing mean and standard error of the mean. It gets tricky with non-parametric statistics because I often see that when people use non-parametric stats. So for two independent groups, typically people will use a Wilcoxon-Mann-Whitney U-test, for instance, and they might even report the medians of the marginal distributions in their papers. But it's not even accurate because in that case, the U statistics is related to the distribution of all pairwise differences. So if you wanted related to it, you could report the median of that, but graphically we need to show something that I've never seen anyone report. So it gets quite complicated. And then if you do a non-parametric type of ANOVA, for instance, I don't even know what you would show in that case.
Okay, and so there's a question here as well about, I don't understand the conflict between non-parametric stats and the use of standard error. The reason for this, not always, but very often when investigators are using non-parametric statistics, it's because they know the sample is not normally distributed or the sample is too small to assess the data distribution. And in both of those cases, the mean and standard error may be misleading. Okay. So we'll have a follow-up question from that, which is what would be the best way to represent non-parametrically analyzed data then? And Guillaume, would you like to answer that?
Well, just probably the best starting point is to plot all your marginal distributions. You know, whatever you measured, show a distribution for each of them, and that'd be a good starting point. Then that, and related to the other question, you really have to understand that each statistical test is a model, and each model is asking a different question about the data. So you need to try to match this graphical representation to the question you're asking. I'll go in depth about this sort of things for quite simple data set in my 2017 papers, but then what I end up doing is essentially I've got two groups and then I make up to 4 graphs showing the same data, but in different ways. So probably there's not going to be one graph that tells the whole story, even for a simple data set.
And if you have too much per chart, it will look cluttered really quickly, right? So that's also, like, from the design principle, a good idea to split sometimes information into several graphs.
Yeah, okay. And I think Helena, the point Helena just made is relevant to another question here. So there's a question that I agree bar graphs are less informative, but sometimes it can be useful to show less info or not, to make things more accessible to the reader when you just want to focus on a specific point. I think one has to be cautious about what one means here because this is science, so we do need to give readers the information needed to critically evaluate and understand the data. If we're making infographics or something else, then it's a bit different. And I think the other principle that's important to understand here is that you can use emphasis and de-emphasis to help draw the reader's eyes to certain things. So often we present things as though it's a dichotomous choice. Like I will see this, I hear this with bar graphs a lot. People will say, well, like, I like the bar graphs because it conveys a clear message. And you know, there's too much information with dot plots, I can't tell what's going on. And so there's actually a specific visualization in the Q&A for this. And I will just share my screen really quickly right now so that you can see this. If you just look at the one in the bottom right-hand corner, this shows how you can use emphasis and de-emphasis to address that problem. So when we emphasize something, we put it, you know, in black or in a color and in the foreground. And when we de-emphasize something, we put it in gray or a lighter tone in the background. So when I look at this graph, my eye immediately picks out, you know, the means or the medians here, and I can see that the third group is higher, but then I still have that additional information that allows me to go back and say, okay, but, you know, what do the distributions look like? How do how are the data distributed, what's the sample size like, and get that critical evaluation of information. And you can do this, the exact same thing, if you're using a combination of, a dot plot with a box plot or a dot plot with a violin plot. You pick one plot that's going to be in black in the foreground, and then the second plot goes in gray in the background. And readers will get the emphasized things first, and then they can go back and review the other thing in more detail. Helena?
Oh yeah, I just wanted to point out, you can go even further, right? In the figure that you showed, it was like you could by increasing the width, for example, of the median, it could get more weight, and then you would be able to see it better. And you could even like make the gray of the dots even lighter or transparent to like de-emphasize them even more, right? And then there's one that you're not showing. What do you think of it? It's called the Death Star, right? Because it looks like a Star Wars fighter thing. It's just the median and the variance.
Mm-hmm.
That's also a possibility if you really, really want to focus only on the center of the data.
Yeah. And I think another place where this overplotting issue comes up, and another common error that I didn't mention, is when you have scatter plots with a higher data density and a lot of overlapping points. So if you have a smaller number of overlapping points, you can make your points semi-transparent, and then regions with overlap will show up darker than regions without overlap. If you have a very high data density, so something like a flow cytometry plot, where even doing that, it's going to be saturated and you won't be able to see the gradation, then you can consider using grid lines or a color-based scale to indicate the density of points in those regions. And obviously, if you're using a color-based scale, then you're using a colorblind safe color map. Okay.
Ta-da!
Yes. Okay, so we'll direct this question to Helena. Should we always write the figure legend title in bold before the figure description?
Oh yeah, well, I like it. I think it's nice if there's a title that I can catch on to and then I see the details below. If you are formatting a paper, for example, for bioRxiv or for your personal use, you can do whatever you like. You can make it in red to stand out or whatever. And if you submit it to a specific journal, then you have to obviously make sure that you are conforming with their requirements. For example, the bold title of a figure, and then everything else in regular text. That's typical for the cell papers and cell group, whatever they're called, and not so much for other journals. So, but they will do the formatting in the end themselves.
Okay, so we have a question for Guillaume here, and it says, since you're using R, do you have any advice on how to make publishing ready figures with any programming language, such as Python, MATLAB, and R? And Helen is excited about this as well. And what would be the standard formatting rules for grid axis size, point size, etc.? And then there's a reply from an audience member noting that in R, there's a package called ggpubr, which to some extent does what the asker describes. So
yeah, big question. So the way I do it is it's painful trial and error. So because there's a problem with when you use a programming language, there is what you set and what you set in your code and what it's going to look like on the screen. But then when you save it to disk as an image and then you then open, you have other problems of rendering and resolution. So quite often I may have different version of the code to render as HTML in a notebook and as versus a PDF or a TIFF that could be used. So try and error. So what I do in R is I use ggplot2, and I've created my own style, which includes all the defaults. So when I create a ggplot object, I load my style and I just automatically format some of the things. And then I just do a lot of copy and pasting. Once I'm happy with the style for a figure, I will just copy and paste the basic formatting so it's consistent across all the figures. The problem really is when you have big busy figures with a lot of panels. What you end up when you save to disk, quite often this problem with overlap, rendering, resolution issues in some part of the figure. So there I need to do some tweaking, save again to disk, check, and then tweak again. I don't have a magic solution for that. And from what I can read in the forums on this different ggplot2 extension and so on, it's not something that's easy to solve really.
Helena, would you like to expand?
Yeah, so this is a discussion that is raging again and again through the data viz world, where I'm it's my biggest home these days. And so for instance, every single data designer that I've met admits to creating something with Python or R and then exporting it as PDF and actually working on it in Illustrator or Inkscape, right? Whichever is available to you, because it's such a pain and you will never know the exact dimensions at the end and you keep on having to go back. So sometimes, so what I personally also do is to just have a certain kind of font, certain kind of size ready in Illustrator, and I just keep on pasting the charts into that. And yeah, that's also how it's still done at most data intensive journalists. For example, Financial Times, we just had a talk from them reporting on COVID, where they have lots and lots of data every single day, and they still do it in vector software at the end of the day.
So yeah, I used to do that when I still use MATLAB a bit, but less and less. When I used to use MATLAB exclusively before I discovered R, I used to have to combine all my panels into Illustrator. But now with R, there's enough packages that let you glue your ggplot2 objects together. And for, at least for my stats projects, where I have more control over the data, obviously, and so on, the figures you see in the papers are all hard-coded in R. I don't touch anything afterwards.
That works if you don't have images that you need to combine it with.
But it's painful.
No, but if you have images and tables, you're getting into the next level.
Yeah, I don't do those.
Just stick to charts.
Yeah. Okay. So Guillaume's advice is don't work with images. And so those of you who are looking at the question box will notice that I am jumping around based on, trying to keep topics as they flow together. We will answer all the questions. So please excuse my lack of any order or structure. Okay. So we have another microscopy question for Helena. Selecting color channels for multicolor microscope images, like for example, if you want to use the Dappy blue example, is it acceptable to use these colors with weaker contrast to highlight the important signal? Also, staying consistent with the colors across all figures is important too, right? So it's the last question, Helena, if you're
Yeah, I'm just rereading it. So I'm trying to understand what exactly is meant. Yeah, I mean, you can, so in the cheat sheet form that we published, Christopher Schmid commented on how to adjust the contrast, and you have to do it linearly for the entire image, but obviously you can do it by channel. You have to do it by channel. So you have to adjust the contrast linearly so that it covers the entire range for the Dappy, and then you have to do it for the GFP, and then you have to do it for the far right or something. You cannot do it at the same time, right? So in that case, obviously it's possible to adjust this, but you cannot do it unlinearly unless there's a very, very, very good reason, and you clearly indicate that in the figure legend for sure. So that's the staying consistent with the colors across the all figures is important. So yeah, I mean, this depends. If you show in figure 1 the control for every subsequent figure, and it's relevant to like be able to compare the Dappy signal across all of the figures, then yes, they obviously have to be treated the same way. It's the same that we say in general for experiments, right? The control has to be treated the same way as the experiment. If that was this, then the answer is definitely yes. But if you have once a Dappy in a cell culture and you have the control right next to it, and then you go to a brain slice and you have a different Dappy signal there, then that's totally fine, because they have different amount of DNA, they have different amount of staining capabilities, etc. So you just have to stay consistent to the control that you're showing. that 6R tool. If that doesn't address it, you can just post it again and I can type.
Okay, so we have another question from Anna. I would like to ask if it's okay to use the bar graphs with a dot, or is it not recommended at all? So this is again part of the Q&A, so I will share my screen yet again. Okay, so my answer to this is always the same. If I would prefer a bar graph with dots over a bar graph any day, but it is always better to just use a dot plot because it gives you everything you need and nothing you don't. There are a variety of reasons for this, of course. So the first thing is when we make bars, we often shade those bars, which obscures the data points that are inside the bars, making them less visible, whereas the ones outside the bar are more visible. The second is that the bars and the vertical lines are actually chart junk. And the thing about bars is they create the illusion of certainty without adding any information. So that bar just looks just as solid and as clear whether you have 2 observations or 2,000 observations. And these are vastly different circumstances in terms of the amount of information we're getting. And so it is an illusion of certainty that we're creating without adding any information. The zone of irrelevance we talked about, if you happen to have one in your graph, then that can lead to a distortion in your perception of the range of observed values. And then lastly, there's something called within the bar bias. And this is where we incorrectly believe that points are more likely to fall within or below the bar than they are to fall above the bar, which is another distortion of our perception of the values. In contrast, if I look at the dot plot, again, everything I need, nothing that I don't. I have clear information about the overlap between groups, where each distribution is centered, whether it's mean or median, and yeah, nothing extra that can confuse or mislead. So that is my answer. Okay. So we have a question for Guillaume. But still the main paper should include a couple of figures, the most important results, and the supplementary materials should include all the additional but important figures, right? It would be a lot of information to put, such as the participant information figures in the main paper.
If you submit your paper to EJN and you pick me as an editor, I will not ask, most of the time I will ask you to move the stuff in supplementary information and put it in the main section. Because quite often people put things that are really crucial at the end. So I realize when you submit to some glamorous journals where they force you to have only very few figures, and I think that's really not doing a good service to science. I think we need more information, and I think it should be out there. The goal of a paper, the results and the methods, is to be able to understand what was done and to let your reader make an informed decision about what they think of the data. So if things are too simplified, you're telling them a story, essentially. You're not giving them enough information for them to make their own mind about your data. So I would think the results section should have much more information than by default than it is common. So I would say put more stuff in there.
Helena?
Would you say that much stuff?
Well, yeah, I know sometimes it can be overkill. Sure, at some point you need to.
The figure that was just posted a couple of hours ago.
That's funny. So we've done it. So for instance, in my line of research, we do quite often single subject analysis. So we do brain imaging, but we have detailed analysis on each participant. So we would have simplified group figures, and then we would show examples of individual subjects, and then we move some of the stuff to supplementary information, where people can look at every participant, and there's a whole full page figure for each participant, for instance. So yes, at some point you need to make some decision, but I would think in most cases it's not the case. People just don't show enough data.
Okay, so your local editor may disagree, so you can send all of your papers to Guillaume, and all of us also support more visualization. So
you'll get lots of them.
Yeah. Okay, so next question regarding proteomic analysis, how can we present big data sets with clear reading information? Because usually there are data that the letters are so small that nobody can read them. And this is from Emanuela. Do we have any proteomics or big data people, Helena?
Well, yeah, big data would be me. Albo, you haven't answered any questions. You want to go? Any suggestions? I can only recommend on the design aspect, really, that you can make a choice, right? Whether to de-emphasize things, and that you have to make a choice of what is the main point, and then you add that text and leave the other out, or you make it in gray to de-emphasize it.
Maybe
Albo.
use the supplementary material to have a huge figure where you can look at everything for the details, for the letters, if you have this kind of big images that cannot fit the usual figure space in a A4 paper.
Okay, anything to add, Guillaume? Or?
Well, I was thinking one of the limitations currently is that we offer mostly static figures. So the future will be dynamic figures where you have access to an online notebook where you can emphasize and de-emphasize different types of data. So you'll be able to explore the data set yourself. Currently, you rely on people sharing their code and the data, and you being willing to go in there and maybe tweak their figures to look at different aspects yourself. It's extremely painful. You might want to do that in specific cases. You really want to know a bit more about the research, but the future will be to have those computational notebooks embedded in the paper. I think some journals are starting to do that, like
eLife.
eLife, yes.
Yeah, eLife, I think, has their reproducible document stack system for doing that. So
I want to try it out and submit a paper.
Yeah. Okay, so a question anonymous attendee would like to know, in some cases, if your data was complex and when you show in dot plots or pairs your data to show the interaction, it would be hard for your readers and the experts to understand. In this case, what do you think? Should I keep it simple or show the data, all the data, to make a clear figure description?
Well, that's a comment I've heard several times, put it in a different way, where people say, well, you know, when you show all those dot plots, it makes my data look noisy. And my answer is, well, your data are noisy. When you simplify, you essentially altering the reality. What you describe with a bar graph or some other type of representation is a representation. It's not the reality. A potential way to go about that would be to have two panels. You have the same figure in different formats. One is simplified, and maybe you start with the general story, so that's the trend and so on. And then you dig deeper, say you switch to the second panel or the second figure and say, this is a reality, that's what's under the hood. And now you can try to describe things in a bit more detail once you've primed your readers with the general structure you want them to pay attention to. So again, the solution is more figures.
I think the other point there is, and this relates to bar graphs as well, people like bar graphs because they make the data look clean. And the reality is that data are messy. And one of the hesitations people have about switching is they say, well, you know, my data are a mess, everyone else's data is perfect. And that's an illusion. Everyone's data are a mess, especially if it's small sample size data. That's just how small sample size data works. Okay. So I can see that Alberto was typing an answer about rain cloud plots. So I am just going to ask him to say what he was typing.
Yeah, so one of the questions was if rain clouds plot were okay, and I think they are. In fact, they merge multiple ways. So they merge in a single figure. You have the violin part of the violin plot, the dot plots, and the box plot. So you have in the same figure 3 different ways to look at the data, and I think it's a kind of Swiss Army knife tool for plotting data points.
just going to show for everyone what they actually look like, right? So that people can get an opinion of what we're talking about. So what I would like to add is from sometimes from teaching workshops and stuff like that, what I notice is that if people are unfamiliar with plots, they tend to be afraid to read on. So if you know your audience and you know the audience is not familiar even with the with a box plot, for example, right? Some audience are really unsure even about that. Then you should maybe avoid going to more advanced or adventurous plots and stick to the simple ones.
Yeah, a couple things to point out about rain cloud plots. First, if you have small sample size data, they are not for you. You need a dot plot. Your summary statistics are only meaningful if you have enough data to summarize, and the box plot and the violin plot that are added require much larger samples than are common in many small sample size animal in vitro studies, as well as small sample size human studies. The second thing, I haven't checked the most recent version of their code. In the early versions, I know that they were using a code that doesn't create a dot plot, it creates a histogram with dots. And so I will once again share my screen just to illustrate what this means. So with a dot plot, you are showing the exact measured value of each point. A histogram with dots is basically grouping the data into bins. So every data point is moved to the center of the bin, and how much movement depends on how many bins. So you can see with a large number of bins, you have relatively little movement and little distortion. With a small number of bins, so it's really important whenever you're using dot plots, whether it's as part of a rain cloud plot or any other thing, check your software. Make sure that it is actually making a dot plot and not a histogram with dots. And the way that you do this is to enter some irregularly spaced data and look at whether or not it comes back as being regularly spaced. Guillaume, do you have opinions on rain cloud plots?
Yeah, not necessarily a big fan, but people got to be careful with the kernel density estimate. So that's the smooth part, because it depends on the algorithm. You'll find entire books on the topic on how to build those, because they used to make inferences about the distribution of the data. And quite often the algorithms don't truncate the distribution. So you might end up with a kernel density estimate that extends beyond possible values. So for instance, you may have negative values where your scores can only be positive. So it might just be a graphic thing, but still it's statistically not really accurate. And again, it shows that, you know, even a graphical representation is some sort of statistical model of your data. So you've got to check what the assumptions of the model are.
Yeah, and you see a related problems with violin plots, which are a form of kernel density plots. And oftentimes there will be a trim tails algorithm or code or option within the code. And that basically allows you to make sure that the distribution does not extend beyond the range of values measured in your data set. Okay, so we have a question about Bayesian statistics from Nico, which I think is a Guillaume thing maybe, and possibly an Alberto thing. So do you think that a Bayesian statistic will become common in neuroscience? Are there some guidelines on graphs to be used in Bayesian statistics?
I like Bayesian stats, but I'm not clever enough to jump completely from frequentist stuff. I can't really answer. I can try to dig up things. So if you use R, there are some really amazing packages to look at the distribution of your posterior distributions and to make some predictive modeling and so on. So I can try to find some resources for that. So yes, well, I don't know if Bayes will become the norm. I would certainly hope so. And yes, they are amazing graphical tools. And I'll go dig some stuff for you.
Alberto, would you like to add anything, or?
Well, as far as I've seen in neuroscience, Bayesian statistics have appeared for a while, and still I've seen very few papers that actually use Bayesian statistics. So I think there is still a long road before it becomes, let's say, more frequent than frequentist statistics.
Maybe after everyone gets a test right, we can move on to that.
Okay, so we have a question from Yao. In my view, the guidelines shown here are also important for transparent data presentation to avoid fraud. Don't you agree? In my peer reviewing analysis, I constantly ask for many of these indications. There are many reasons why one can benefit from seeing the data, in part for critical evaluation, in part for making sure things look the way they should, in part for being able to reproduce things in our own lab and knowing what we should expect and what things should look like. Bar graphs and other graphs that only show summary statistics hide many things that are really important information for scientists to have. In terms of peer reviewing, I also always ask for all of these things. And so I think one of the things that I do as a peer reviewer or as an editor is to provide papers that are easily accessible to help people understand why it is that I'm saying this and why it is that I'm recommending something, and also just what better practice looks like to make it easier for them to actually change what it is that they're doing. And I think those types of citations can be very informative for authors, and they also increase the likelihood that the authors will actually go ahead and implement changes. Do others have comments on that?
No, I think it's fine.
Okay, so we have a question from Sarah about how to represent data from several participants that is not continuous. For example, Likert scales. Any ordinal data people?
Yeah, interesting. Yeah, I just had a PhD student working on a paper on Likert scales, but doing some simulations of the ratings.
I'm
for this
Googling what is a Likert scale.
Well, you got 5 or 7 points, and you ask people a question, and you go from one extreme to the other. It's a sort of
like
a questionnaire. Yeah.
Yeah, like the agree to disagree, or agree, strongly disagree.
Oh, I never know what to put there. I always put the middle because I can't be bothered.
Those are they're really tricky because they typically they're presented using statistical methods that are completely inappropriate. Because the Likert scales, because they're ordinal, you can't use mean and standard deviations. You need some proper modeling that takes into account the ordinal nature, and that you have some continuous distribution that is in people's head that maps onto those scales. So you can't really show this type of data efficiently. The best way is to share your data and show the outcome of some of the modeling. I've got.
But in principle, you could just make it like a heat map, no? If you just, because you just take the counts, you don't need to make statistics on it. Just present the counts, or
you can make some histograms for the different bins. Yeah. Yes. But then if you want to have like the summary statistics, that becomes tricky because you can't just use the regular ones, because the assumptions of your classic test are all violated by the Likert scale.
Okay, so histograms are better than summary statistics. Summary statistics are not good for ordinal data.
Yeah, you could produce summary statistics, but from a computational model that takes the ordinal data, the nature of the data into account.
Wow, I learned something myself now.
I will send you a link to a preprint about that has some nice illustrations of the problem.
Okay, so there's also a question that is split over 2 questions. So this is regarding the dot plots with the highlighted horizontal bars. In this case, is it fine not to show the standard error or the standard deviation? Again, summary statistics are only meaningful when you have enough data to summarize. So if your sample size is really small, like if you have 3 or 5 observations, your standard error or standard deviation are not particularly meaningful, and you are better to just show the dots and the median. That's my preference. Do others have a different opinion on that?
So showing what, the median? And
yeah, so I had showed some dot plots of small sample size data where I did not show summary statistics. I only showed a median line, and there were, ends of 5. And so the question was, if you are showing a dot plot, is it okay not to show the standard error or the standard deviation?
yeah, absolutely. I mean, you make assumption about the data, and you asking a specific question when you show the standard deviation or the standard error. There might be other statistics that are more useful. Or if you have very few points, it's hopeless, because any summary statistics will be heavily misleading. So just show the dots. I mean, the median is a good starting point, because
yeah.
But for a small sample size, the median might be an allocation where there's no points at all.
Okay, so small sample size, yes, you can definitely not show standard error, standard deviation. You may or may not want to show a median. With larger sample size, I think you have more information about the data distribution, and so summary statistics become more useful and more relevant to see. Okay, I have seen people presenting box plots for parametrics tests, like a t-test, and in the results section, it's written as mean and standard deviation, and in the figure legend, it's median with a range. With the scatter plot, can we avoid this, or some other graph like a dot plot?
Well, it goes back to the link between the question you ask and the graphical representation. So if you make inferences about the medians, show the medians, or you might want to show the medians and the mean to assess the. It's a non-parametric measure of the skewness in your data. It's useful. Otherwise, I don't know. It depends what you make with it. Sometimes I think people are asked to make better figures, so they just add stuff asked by the reviewers or the crazy editor, but then there's no link to their analysis and their inferences. And then that would just leave the readers confused. So I think you need to try to match and think more deeply about what that means. The worst, I think, is if you put stuff in a graph and don't talk about it. Let's say you superimpose the median and the mean, and you just don't explain anything. You just say it's there. Try to build a narrative around that. What do you learn from it? Try to describe that. And then you might realize, as you provide a description, that when comes the inferential part with the statistical test, you realize, well, actually, I'm not asking the right question there. I should probably ask a different question using a different statistical test.
So I think the one thing that it is important to consider here, and the one reason why some people will do this, is if someone uses your data for a meta-analysis later, the meta-analysis statistics are really heavily built around means and standard deviations and parametric summary statistics. So if you are using box plots or whatever along with parametric tests, so your figure is showing the full distribution, but your tests are based on, the parametric tests as the user is specified, and that those tests, the distributional assumptions are appropriate, then it can be helpful to report those things so that if someone wants to use your meta-analysis, your data for a meta-analysis later, they have the ability to do that without trying to figure out, the summary statistics that basically aren't shown on your graph. The problem is when people present those summary statistics for distributions that clearly aren't normal, and then the summary statistics make no sense. That's a larger issue. So,
and the best way to be future-proof, so that if someone then suddenly decides to do meta-analysis on something else, is to actually share your data so that people can do whatever they want.
Yes, and then they can do an individual patient data meta-analysis, which is much more useful. Alberto?
Yeah, I completely agree. That the best way is to have the raw data set, so all information are there, and then yourself, if you want in the future to reanalyze those data, you can do it.
Okay, we have run out of questions. Are there more questions? Or is there anyone who would like to ask a question as a human with a voice?
That would be nice. Don't know if there's some stuff in the chat. Well, thank you for all the questions. It's all very stimulating.
Helena has promised that we will stay until midnight if that's what it takes to answer all the questions. So,
yeah, I need someone to bring me a snack, though.
Alberto will bring you a snack. Okay, so if I'm making a meta-analysis and in some cases I end up with a few points, say 3, should I report the mean and standard deviation or try another type of plot? If you have 3 points, so is this for the dot plot or the original graph? If the original data set that you're using for your meta-analysis has 3 points, then a dot plot would be good. If data aren't available and your data are in a dot plot, someone doing a meta-analysis can use something like Plot Digitizer to recreate your data set and calculate the mean and standard deviation. That is an option with dot plots of small sample size data sets that you wouldn't be able to do that with other things. But it would be an option in that case. So yeah, I still generally favor, if you have really small sample size stuff, just leave the summary statistics out of it, because there's a lot of potential for misuse by others later.
I didn't know you could read data from a plot.
Plot Digitizer is a very useful tool, and everyone who uses meta-analysis does
Be careful, it's very addictive.
You can go back to 100-year-old papers.
Yeah, it's very useful when you teach stats and you want examples from a figure.
Nice. Well, can you like post that in the chat? Digitizer, what's it called?
Plot Digitizer.
Wow.
Yeah, you load up the figure. It's also very useful when you want to, well, for methods papers, when you want to reanalyze data sets to see if different methods would bring different results.
Okay, are there other questions?
Not anymore, huh?
Okay, no, we have one more. I often get tied up thinking about what is my N. For example, X mitochondria quantified from Y cells with Z independent cell preps. Any advice? There is an excellent paper in PLOS Biology called What is N in Animal and Cell Culture Experiments? I think that's what it's called. That is an excellent resource to start with for thinking about what are your biological units versus your experimental units. I would start there. Others suggestions?
I think that's what an issue a lot of people struggle with. Every year I'd have, for the neuroscience master program, I do the vivas, and it's not my field, but I end up asking the question, what are the units of measurement? Because they've got scatter plots sometimes that don't match with the number of animals that were used. So it's unclear. I would be dealing with slices, cells. The breakdown is often quite unclear. And when people are put on the spot, they just don't know. So I think it is a very common problem. I think the best way is to be transparent about exactly what was done. So there might be more details in the methods section about measurements, number of animals, number of slices, and what goes in the graphs. And when people put scatter plots, quite often that's where you realize there's a mismatch between the analysis and the structure of the data, where people might combine things into a correlation analysis where there's multiple measurements per animal, where you would need a mixed effect model, for instance, but everything was pulled together as if all the points were independent.
Yeah, so
this is something I think that should be discussed more among scientists, right? So these kind of questions should probably be discussed openly within a group seminar, etc. And I feel like that's rarely the case.
But a different tradition in psychology, it's much more clear-cut, because you have a certain number of participants, and then you have a certain number of conditions, and for each condition you have a certain number of trials. and people are really trained to think about that. So there it's quite clear cut, even though in the literature it's not necessarily that clear in the method section. But when it comes to animal studies, I don't know why, it must be the training is different, where it's often really unclear how many degrees of freedom you have at different levels of analysis.
Okay, so I posted the paper that I mentioned in the chat as well as in the answer to the question, and you can also look for resources on pseudo-replication is typically what this phenomenon is called, and there's a lot of literature examining it as well. So I think we have answered all the questions, and so it's time to close the webinar. So I'd like to thank all of you for joining us today. And in such a short time, as I mentioned, we can really only cover specific things. So we have provided you with a lot of resources in the website for the webinar, and I would encourage everyone to go ahead and take a look at those resources list and go ahead and review anything in more detail that you think might be interesting to you or relevant to your research. And please don't hesitate to contact us via Twitter or email or however you prefer if you have further questions that occur to you after the webinar. So thanks again to all our panelists for excellent presentations today, and to all our attendees for wonderful questions.
And thank you to our moderator, Tracey Weissgerber, and her talk.
All right, thank you, everyone.
Thank you. Bye, everyone.
Bye.